Hi, everybody. Okay, in this video, we are going to take a look at Chapter 9. In Unit 9, we are taking a look at what is called antimicrobials, which is, again, if you think microbials are microorganisms, anti means how we counteract those. So when these microorganisms are colonizing in our body and causing infection, how do we stop that from happening? Well, that's what we're going to take a look at. So let's go ahead and look at Chapter 9 then. Chapter 9, if you're following along in your textbook, it's chapter 9, no matter which, edition of the book you guys have. So let's see here. Okay, here we go. So chapter 9, controlling microbial growth in vivo using antimicrobial agents. Excuse me. Okay. So in this chapter, we want to take a look at the characteristics of an ideal antimicrobial agent, how they work, and then we'll kind of break them down. And I've cut this chapter up a little bit to focus on what we focus on, like I've mentioned before, bacteria, viruses, and fungi. So we're going to look at those antibacterial, antifungal. We don't really take a look at antiprotozoal, antiviral, though. We'll see what I mean by drug resistance, some strategies in the war against drug resistance. And then I think we've skipped over empiric therapy and some undesirable effects. We will take a look at those. Okay. So our introduction here. So chemotherapy, you guys have heard that term before, oftentimes used with cancer patients. That term is used to describe the use of any chemical or drug to treat any disease or condition. So a chemotherapeutic agent is any drug to treat any condition or disease. So that's kind of a broad term talking about, the type of drug that's used to treat anything. Remember, not all diseases are pathogenic diseases caused by a microorganism. That's where we're going to narrow this down a little bit. So an antimicrobial agent is more specific. An antimicrobial agent is now referring to a chemical or a drug that is used to treat an infectious disease. Again, an infectious disease caused by an infection of a pathogen. So either how this works, we're either going to inhibit, so stop the growth of that pathogen, or killing that pathogen in vivo, so meaning inside the patient's body. Some antimicrobial agents are referred to as antibiotics. But as we've mentioned before, antibiotics are typically prescribed for bacterial infections only. Okay, so drugs used to treat, so again, breaking that down a little bit further, when we talk about these antimicrobial agents, drugs used to treat any bacterial disease is going to be referred to as an antibacterial. Those used to treat fungal diseases is going to be called an antifungal. Makes sense. Those used to treat protozoal are called antiprotozoal, antiprotozoal, and those that are used to treat viral are called antiviral agents. An antibiotic is a substance produced by a microorganism that kills or inhibits the growth of other microorganisms. Typically, again, as I mentioned, they're focused on bacterial microorganisms. Antibiotics that have been chemically modified to kill a wider variety of pathogens and reduce the side effects, meaning that it's not specifically targeting one particular type of bacteria. This is called a semi-synthetic antibiotic because it's something that has been worked with to try and target a wider variety. And we're going to talk a little bit about the term broad spectrum a little bit. You guys may have heard of that when you guys are taught, when doctors prescribe a broad spectrum antibiotic, it's typically for, again, a variety of different types of bacteria, not just one type. So that's what we'll take a look at. So that includes semi-synthetic penicillin, such as ampicillin and carbonicillin. You guys maybe have heard of, especially ampicillin. This, I think we've talked briefly about this before. This is the discovery of penicillin by Alexander Fleming. He was the one who came up with the penicillin. What he had was colonies of Staph aureus growing. This is, again, Staph aureus. If you guys think of the term Staphylococcus, we're talking about bacteria in clusters and the round-shaped bacteria. So Staphylococcus aureus is growing here in this Petri dish. You can see by pointing out letter A there, that's what all these little circles are. Colonies are poorly developed in the area of the plate where the antibiotic was. So here's what letter B is talking about. So this entire Petri dish was covered with that Staph aureus. But what happened is letter C, this is a mold that was dropped onto this Petri dish, specifically Penicillium notatum, which is a mold. And what happened was that around that particular mold, you can see the bacteria is starting to die off. And that's where Alexander Fleming said, wait, we might have something here. This is a big deal. We can kill off Staph aureus by using a mold. And of course, ideally, we don't want that mold to be something that's harmful to patients. So that was studied. And in turn, we figured out that it was not. This is going to be one of those molds that is okay for patients to take. And it eventually led to the discovery of the antibiotic penicillin. So, and ideally, so as I just mentioned, ideally, an antimicrobial agent will do this. It's either going to kill or inhibit the growth of that pathogen. In order for it to be effective, we want to make sure that it stops that pathogen from causing destruction. We want it to definitely cause no damage to the host. We don't want it to make the host sick. So we don't want there to be, so a big thing when we say damage, that's why we have to ask if a patient is allergic to anything, because we don't want it to cause any allergic reaction to the host, because then we're trying to stop a pathogen, but then we're just causing a whole different problem. We don't want that to happen. We want it to be stable when stored in solid or liquid form. We want that medication to be able to be in pill form or liquid form that the patient can take, and it not, be disrupted in any way. We want it to remain in specific tissues in the body long enough in order for it to be effective. We don't want it to be something that the body just processes immediately, digests, and gets rid of. Instead, we want it to be something that stays within the body so that it can do its job. And we want it to stay long enough so that it can do what it needs to do by killing or inhibiting the growth of the pathogens. And then again, we want it to kill the pathogens before they mutate and become resistant. If you guys have been exposed to this viruses, bacteria, sometimes they become resistant to antimicrobial agents. They kind of learn and discover what's being targeted against them, and they can they mutate, change their shape so that they can't be recognized. There's different things that they can do to mutate, and then these antimicrobials are ineffective against these. So we don't want that to happen either. So the 5 most common mechanisms of action for an antimicrobial would be a couple different things. Some types of antimicrobial will inhibit the cell wall synthesis, which means that cell wall does not grow around that bacteria, and therefore it doesn't work as properly. So therefore it's not going to be able to spread as it would. Damage to the cell membranes, very similar, causing damage to that outside envelope. It'll inhibit the nucleic acid synthesis. Nucleic acids, remember, is that genetic information. Remember, viruses can be either DNA or RNA. Bacteria have, very similar genetic information. So if we stop that particular microorganism from knowing what it needs to do in order to spread infection. So that an antimicrobial sometimes works that way. Sometimes it inhibits the protein synthesis. Proteins are what is built. If maybe you remember from viruses, how viruses work is they can inject either their DNA or their entire virus goes into the host cell, and the host cell will grab the what they use to help to I'm trying to explain this a little bit better. What they use in order to replicate themselves, their own cells, they use what's called these, they're kind of a copying machine. And it takes these proteins and creates more. And what it does, a virus will use those copying machines in the cell and copy itself and copy the virus so that the virus then spreads. But if we stop that growth of the protein, then we stop that virus from becoming efficient. So stopping the protein synthesis or inhibiting the enzyme activity. Remember, some bacteria produce enzymes or exoenzymes that the enzymes themselves cause harm to the host, and stopping that would stop that damage. So antimicrobials work in various ways, and it's one of these mechanisms that they will use to stop the growth of that pathogen. So antibacterial, the main thing I want you guys to be familiar with on these antibacterials, again, are used to target bacterial cells. So again, bacterial infections are going to be stopped by antibacterial agents. So you don't need to specifically know the specific types that destroy gram-negative versus gram-positive. But I do want you guys to know the difference between narrow spectrum and broad spectrum. Whereas, as I was kind of briefly mentioning before, that broad spectrum antibiotics are prescribed for, and you can see ampicillin, as we were previously talking about those semi-synthetic uh, agents, that ampicillin falls into that category, as well as this broad spectrum antibiotic, because it is created in order to kill off a variety of different types of bacteria. Sometimes bacteria, the best way to kill it, we prescribe, just like Alexander Fleming grew that on a petri dish. We look to see which antibiotic is going to be more effective against that specific bacteria. That's going to be where that narrow spectrum comes in, where it's only going to target these certain types of bacteria. But broad spectrum means it's more broad, that this particular antibacterial agent can kill this type of bacteria as well as this type and this type and this type. So that's the big thing, that antibiotics that are destructive to not just the gram-negative bacteria, but both gram-positive and gram-negative, are called broad spectrum, because it falls into more of a broader category. So that's kind of the only thing I want you guys to know about that the difference between narrow spectrum, meaning it's just kind of a narrow range, only targets a specific type of bacteria. Broad spectrum, a more broader variety of bacteria. Multidrug therapy, sometimes one drug is not going to be sufficient, so 2 or more drugs may be given simultaneously, such as in the treatment of TB, tuberculosis. Most of the time, those patients will need to receive a couple different medications. And that's, as that name implies, multidrugs are given. So when we use multidrugs together, when we're using not just one medication, but 2 or more, then here's where we need to focus on synergism versus antagonism. When 2 things work in synergy, that means they work very well together. Whereas if 2 individuals oppose each other, now this is now talking about medications, but also individuals as well. When people oppose each other, they're called antagonists. They're opposite of one another. Synergism is good in terms of medication, antagonism is bad. And that's what we're talking about here. Synergism takes place when 2 antimicrobial agents are used together, and they can produce a degree of pathogen killing that is greater than if they were just using one drug alone. So meaning that if a patient came in and needs a particular medication and we prescribe them one antibiotic, it may not be as effective. But when we use multi-drug therapy, sometimes 2 drugs used together target that bacteria even better, and then it kills off that bacteria even faster. That is synergism, when they work very well together. So again, that's a good thing. Antagonism, on the other hand, is a bad thing. We don't want this to happen. And sometimes certain medications fall into this category. They cannot be given at the same time because they actually work against each other. The extent of pathogen killing is going to be less than if that one drug was used by itself. So that's something that we study when we study pharmacology. You guys will get into that and see how drugs sometimes can counteract one another and should not be prescribed together because they oppose each other. So that is synergism versus antagonism. All right. Antifungals. So again, big thing to focus on here. Antifungals target fungal infections. So as far as, how they're targeting, what they're targeting, I wouldn't worry about these, how they either bind with cell membrane sterols, or they interfere with the sterol synthesis, or they block the replication process of either mitosis or nucleic acid synthesis. That they, again, the big thing I really want you guys to just know about this, focus on just how antifungals are used to target fungal infections. That's the big thing from that. Don't worry about specifics. So let's go ahead and move to the next one. So antivirals, as we've already kind of mentioned this before in our virus chapter, antiviral agents are the newest weapons in antimicrobial methodology, because a lot of times, again, what we've studied with viruses, because they're so various as far as their shape, their size, whether they have envelopes or not, or capsids, or made up of capsomere, it's just all kinds of different types of viruses. So sometimes viruses are hard to treat. But these are a little bit newer. As we've studied more antimicrobial agents, we've come up with some that have been effective. Again, sometimes antivirals are difficult to develop because viruses are produced within host cells, so it's hard to really target those viruses. Some drugs have been developed that are effective in certain viral infections, but not in others. They typically work by inhibiting the viral replication in the cells. And antiviral cocktails, just like we talked about with the antibacterials, the multidrug therapy, sometimes a variety of medications, several different drugs are administered simultaneously. And that's referred to as that antiviral agent cocktails, and those are being used to treat HIV. So again, antivirals are pretty new. We know again that they are targeting viruses specifically. Okay, drug resistance. When I refer to the term superbug, so earlier I'd mentioned what happens, how sometimes microorganisms have the ability to mutate. They almost learn what these antimicrobials look like, and they figure out how they can avoid being targeted by these antimicrobials. They mutate, and that allows them to become resistant. And that's what a superbug is. So superbugs are microbes, mainly bacteria, that have developed this resistance, that have become resistant to one or more of these antimicrobial agents. Infections that are superbugs are incredibly difficult to treat. And for that reason, that they have been able to change their appearance so that now scientists are going, well, we need to come up with a different one now, because now this antibacterial is ineffective against this particular bacteria because it has changed now, when we need it to be specifically something that targets what was the previous shape. And that's it. Like I said, that sometimes it's a mutation, it's just a different things take place. So here are some different bacteria that you guys may have heard of that have been able to resist antibiotics. And again, constantly studying these and figuring out what we can do to make sure that these can be targeted. So that's what a superbug is. So that's what I want you guys to know from this slide. You don't have to worry about knowing a specific superbug, but know what a superbug is. Okay, so how do they become resistant? So I've kind of briefly mentioned that already. Some bacteria are naturally resistant because they lack the specific target site for the drug, or the drug is unable to cross the organism's cell wall. So there's a couple different ways that this takes place. That the bacteria hasn't really done anything. It's just this specific type of bacteria doesn't have that target site that the drug is looking for. If it sometimes it just doesn't have the ability, because bacteria, if you remember, sometimes bacteria have capsules or cell walls around it that keep it protected. And sometimes they're unable to penetrate through that cell wall or cell membrane. So it cannot reach the site of action where it's going to cause the destruction to that bacteria. So this particular type of resistance is called intrinsic resistance, meaning that it already exists within that bacteria. Whereas if it does the mutation that I was mentioning to you guys, if a bacteria that were once susceptible to a particular drug and has since become resistant, this is now called acquired resistance. It developed this resistance after the fact. Okay, so I do want you guys to know the difference between intrinsic resistance, which means some bacteria just already possess this resistance, and then acquired resistance, which means it develops that resistance after being exposed to that drug. Very similar to how our body's immune system works, that oftentimes we have to be exposed to a certain virus in order to learn what it looks like. And then our body can produce antibodies in response to it after being exposed to it a second, third, and fourth time. Bacteria works a very, very similar way. So this just talks about what I mean by that. So before a drug enters into a bacterial cell, it must first bind to a protein on the surface. So that's what I mean when I was talking about the target site. It must bind to a protein on the surface of that cell, and these are called the drug binding sites. If again that binding site doesn't exist, then that drug cannot attach to it and therefore cannot get into that bacterial cell. So sometimes a chromosomal mutation affects the surface of that binding site, and it can prevent that from taking place. And that results in that drug resistance. To enter into that bacterial cell, a drug must be able to, so again, what I was also referring to, that if a drug cannot cross the cell wall or cell membrane, it may not be able to reach that site of action. So to enter into that bacterial cell, it must be able to pass through that cell wall and cell membrane. And sometimes it develops that mutation that allows it to alter the structure of that cell membrane, therefore causing kind of a blockage for that drug. And therefore that drug cannot enter the cell. And that also will lead to drug resistance. Bacteria can also develop ability to produce an enzyme that destroys or inactivates the drug. Bacteria, remember, that's one of its defense mechanisms that and that's how it causes destruction within our body. But it can also do the same thing to drugs as well, that it can develop these enzymes, and the enzyme will destroy or inactivate the drug itself. Many bacteria have become resistant to penicillin because they have acquired the gene for that specific type, that the penicillinase, which is that production during conjugation. They have been able to learn what penicillin looks like and how they can destroy it. Okay. And then in bacteria, plasmid that contains multiple genes for drug resistance is known as resistance factor, R factor. Okay, so drug resistance is bad. We don't want this to happen. But this is how this works. Another way that this happens, bacteria can also become resistant to drugs by developing the ability to produce multi-drug resistance, multi-drug resistance pumps, also known as MDR transporters or efflux pumps. So meaning, as it mentions here in the description, an MDR pump, meaning this bacteria has developed this mechanism that once the drug adheres to the bacteria, what it can do is it causes that cell to pump out drugs before they can damage or kill the cell. So it kind of ejects them. It gets rid of that bacteria. So even though it may be able to attach or get into the cell, it has developed this mechanism that pumps it out before it can do any work. So in summary, bacteria can acquire resistance to these antimicrobial agents by chromosomal mutation or by acquisition of a new gene by what's called transduction. Not that you have to know that term. That just means an acquisition of a new gene, transformation of that gene, and most commonly by conjugation. So these are different ways that but bacteria can change their genetic information in order to try and either hide from drugs, get rid of those drugs, or kill off those drugs before the drugs can cause any damage to them. So that's how this drug resistance works in this just variety of different ways. So this just shows, again, these different mechanisms by which bacteria can become resistant to these antimicrobial agents. So what are some things that we can do? How can we stop this? One, education of healthcare professionals and patients. We need to understand how this works. It's important that we know that this is possible. Patients should also stop demanding antibiotics every time they or their child is sick, because oftentimes it's the over-prescribing of antibiotics that it's almost like by doing so, we are just constantly teaching bacteria what these antibiotics look like, even though they may not need those antibiotics. And just like when we are constantly exposed to different viruses, our immune system just becomes better. We acquire more resistance because we are constantly exposed to it. And that's what's happening when we constantly expose a bacteria to an antibiotic. So physicians, in the same aspect, physicians should not be pressured by patients and should prescribe only drugs when necessary, when warranted. Clinicians should really focus on prescribing those narrow spectrum drugs if lab results indicate that it's going to kill that pathogen that's causing harm to the patient. Because again, those broad spectrum drugs are really used only when we need to target a variety of different things. Maybe we're unsure which antibiotic is going to, kill off this drug. But if we know that a very specific narrow spectrum drug is going to kill it, then we need to prescribe that instead of constantly giving patients these broad spectrum antibiotics. Patients should destroy any excess or outdated medications. Again, just taking them at will. All we're doing is just exposing our body more to these antibiotics and teaching those bacterial cells what they look like. Antibiotics should not be used in a prophylactic manner, should not be used in any manner other than what's indicated. Healthcare professionals should practice good infection control, constantly making sure that we're not exposing our patients to new pathogens, so that again, we're having to prescribe these these antimicrobials all the time. We want to keep them from developing these, especially these drug-resistant bacteria. And patients should take drugs in a manner that is prescribed. Most of the time, and you guys know if you've ever been prescribed antibiotics, you were told that you need to take the whole regimen. Even if you're halfway through and you start feeling better, you still need to make sure that you kill off that bacteria completely before it starts just learning what that antibiotic looks like without being exposed more to that antibiotic to kill it off. We need to make sure that we take it until it's completed. Okay. All right, guys. So that is chapter 9. So looking at our antimicrobial agents and how they target these infectious pathogens. So if you guys have any questions at all, you guys know, just let me know.